Hello, my name is Matvei Khoroshkin, and I want to tell you about the variability of B vitamin dependencies in human microbiome genomes. Firstly, a brief introduction. B vitamins are biochemical cofactors that are essential for any living systems. B vitamin variability can influence structure of any microbial community. In particular, human microbiome is a medically important microbial community. It can influence the human's health, it can regulate immune response and fat storage of human. It depends on host genetics and diet and some other factors. So fundamental question is, how is human microbiome structure influenced by B vitamin availability? A brief about our results. We established a new method for B vitamin phenotype prediction in bacteria using whole genome sequences as an input. The method was applied to predict phenotypes for 8 B vitamins in more than 1,000 reference genomes from human microbiome project. The predicted phenotypes were used to evaluate the B vitamin prototroph-auxotroph ratio in real metagenome samples of the human gut microbiome. Our reference genome set contains more than 1,000 genomes from 7 phyla, the 4 main phyla and 3 residual. Reconstruction of B vitamin biosynthesis pathways in reference genomes were made using the subsystem-based approach that is implemented in seed server. Subsystem is a generalization of the pathway term. Each subsystem includes functional roles mapped to genomes. So subsystem allow you to know the presence or absence of selected functional roles for any genome you want. Seed is a genomic tool that implements subsystem-based approach and provides accurate gene functional annotations. These annotations are highly more exact than annotation made by traditional gene-by-gene functional annotation method. This subsystem approach is highly efficient for analysis and annotation of large sets of genomes. Using our predictions using our reconstructions of biosynthetic pathway, we developed phenotype rules for 8 B vitamins. Phenotype rules, prototrophs are organisms that are capable to synthesize some vitamin, and auxotrophs are organisms that can't do it. So this slide shows the simple example of the biosynthetic pathway. It's biosynthetic pathway of folate, and it consists of a few sequential reactions, and one of intermediate compounds can be transported into the cell. So downstream genes are required for both prototrophic and auxotrophic organisms, and the unique property of prototrophic organisms is the presence of upstream genes. So in this case, the rule is simple. If a genome contains upstream genes, it's a prototrophic genome. The rules like that were developed for all investigated B vitamins. Here they are shown schemes of biosynthetic pathways for 8 B vitamins. We were interested in does it exist some taxonomic level that divide all the bacteria to groups, that 2 organisms from the same group have the same phenotypic pattern. Our genome set contains more than 100 species containing more than 1 strain, and 17% of them show variability of vitamin phenotypes among the strains, but only less than 4% show variability of phenotype for more than 1 vitamin. So we can make an assumption that species level divide the bacteria to conservative by phenotype groups. And higher taxonomic levels show variability within their organisms, within their groups. So more than 60% of genera containing more than 1 species or strains show variable vitamin phenotypes among their organisms. Investigated genomes were grouped by body site they were extracted, and we computed the B vitamin phenotypic patterns as arrays of prototroph and auxotroph ratio for each body site. You can see that distinct body sites show diverse distribution of B vitamin dependencies. For example, gastrointestinal tract and skin are populated mostly by prototrophs, and oral tract and urogenital tracts by auxotrophs. It is surprising that oral cavity and urogenital tract have B vitamin phenotypic patterns that are similar more to each other than to gastrointestinal tract. Investigated organisms were grouped also by the taxonomy. Here's a phylogenetic tree of the most abundant genera of the most abundant orders from our genome set. Their names are colored by films. So different taxa show diverse distribution of B vitamin dependencies. So proteobacteria are mostly capable to synthesize all the B vitamins except cobalamin. Bacteroides too, and for example, firmicutes are mostly prototrophs for all the B vitamins. Our prediction were used to estimate auxotroph-prototroph ratio in real human gut microbiome samples. In the last few decades, a lot of studies of estimation of phylogenetic diversity in metagenome samples using the 16S rRNA gene amplified sequencing were published. So data from these studies provide clusters of similar sequences, which named OTUs, it means operational taxonomic units, with their abundances. Assumption that sequences from the same cluster belong to the same species allow to calculate a prototrophic and auxotrophic organism number in each metagenome sample. So we evaluate the prototroph-auxotroph ratio values in more than 500 of human gut microbiome samples using these data. Microbial diversity of all the analyzed samples are well covered by our reference genome set. It is a good marker of good quality of our reference genome set. Obtained values allowed us to estimate variability of prototroph-auxotroph ratio in human gut microbiome. And here it is shown. Each histogram shows distribution of values of percent of auxotrophs in all investigated samples. So for few vitamins like thiamine, riboflavin, niacin, and folate, you can see that most samples have a low percent of auxotrophs. It means that human gut microbiome is populated mostly by prototrophs for these vitamins. On the contrary, cobalamin, for example, show the high percent of auxotrophs. Few more observations were done from analysis of prototroph-auxotroph ratio in microbiome samples is the interindividual variability of auxotroph-prototroph ratio is much higher than temporal variability, and for any vitamin, no significant shift of auxotrophy level associated with host diet changes was found. So conclusions, we developed a new genome-based method for B vitamin phenotype prediction in bacteria which can be automated. We applied this method to predict phenotypes for 8 B vitamins in more than 1,000 reference genomes from Human Microbiome Project. The predicted phenotypes are applicable to evaluate the B vitamin prototroph-auxotroph ratio in real metagenome samples, and we estimated variability of prototroph-auxotroph ratio in more than 500 of metagenomic samples of human gut microbiome. This work was made in collaboration with Dmitry Rodionov and Andrei Osterman and a few other researchers from Sanford Burnham Medical Research Institute. Thank you.
Thank you. Any questions? Okay, thank you very much.